Welcome. In this video we'll walk through how to choose a genomics experiment. Bulk versus single cell, RNA versus ATAC, short versus long read, and spatial. The goal is simple, to match the method to your biological question. Let's start with the big picture. Modern genomics is really a menu of assays, and it's easy to reach for whatever is newest or highest resolution. But the best method is the one whose strengths fit your question. 3 axes organize the choices. Which molecule you measure, RNA or chromatin, at what resolution, a population, a single cell, or a position in tissue, and what read length for extra modality you add. So before choosing, ask 3 questions. First, what do you actually want to measure, expression, regulation, isoforms, or location? Second, at what resolution, a whole population, each individual cell, or a place within the tissue? And third, do you need a particular read length, or a second layer of information? Answer these 3, and the assay usually chooses itself. Let's begin with bulk RNA sequencing. It pulls transcripts from every cell in the sample and reports one averaged expression value per gene. It's cheap, inexpensive, and very robust. The trade-off is that it cannot tell you which cells produced the signal. That single averaged number can hide very different underlying biology. So, when is bulk RNA the right call? When your cells are already sorted, or the tissue is fairly homogeneous. When you're comparing many samples or conditions, like a clinical cohort or a time course. When you want tissue-level differential expression. When maximum depth per sample on a limited budget matters. And when cell type resolution simply isn't the question. Single-cell RNA sequencing barcodes each cell, turning that one average into a matrix of cells by genes. Suddenly heterogeneity becomes visible. You can see cell types and states, detect rare populations even at low frequency, and measure both the proportions of cell types and the trajectories cells follow as they differentiate. Reach for single-cell RNA when the tissue is a mix of many cell types. When you're discovering cell types or rare populations. When you need to separate a shift in composition from a change in each cell state, a distinction bulk cannot make. When you're mapping trajectories. Or when you're building a cell atlas. Putting them side by side, bulk gives one number per gene, with composition and expression entangled, and it's ideal for deep, cheap comparisons across many samples. Single cell gives a profile per cell, separates composition from state, and reveals rare and novel types. They're complementary, not competing. Bulk to scale, single cell to resolve. Within single-cell RNA, most experiments use short reads. These tag just one end of each transcript, about 100 bases, and count genes. The result is gene-level counts, very high throughput, low cost, and a mature analysis ecosystem. For most single-cell work, and especially for building atlases, short reads are the workhorse. Long-read single-cell RNA instead sequences the entire transcript. That lets you see structure a 3' count cannot, which isoform is expressed, not just which gene, the exact start and end sites of the transcript, and fusion transcripts or allele-specific sequence along the whole molecule. So how do you choose read length? Short reads are cheapest and highest throughput, and give gene-level counts, ideal for cell atlases. Long reads resolve full transcript structure, isoforms, start and end sites, and fusions, at a higher cost per read. And remember, about 95% of multi-exon human genes are alternatively spliced. If isoforms matter, go long. Next, spatial transcriptomics. These methods keep the location of each measurement, so you learn where cells sit, not just which cells exist. There are 2 broad families. Imaging-based methods, like xenium and 𝗫𝗫, give subcellular resolution but measure a targeted panel of genes. Sequencing-based methods, like Visium and Visium HD, capture the whole transcriptome, but spot or bin resolution. Reach for spatial when tissue architecture is part of the biology. When you care about cellular niches and neighborhoods. When you want to study how cells interact in their native context. When where cells are matters, not only which cells are present. And when you need to place your cell types back into the tissue. The central spatial trade-off is resolution versus coverage. Imaging gives you subcellular precision, but only for a limited, pre-chosen gene panel, best when you already know your targets. Sequencing captures the whole transcriptome, but coarser resolution, best for open-ended discovery. Your question decides which side of that trade-off to take. Now let's switch molecules. Where RNA tells you what a cell is expressing, ATAC tells you which regulatory DNA is open. Single-cell ATAC profiles open chromatin per cell, revealing the promoters and enhancers in use, mapping regulation to specific cell types, and hinting at which transcription factors are active. As with RNA, ATAC comes in bulk and single-cell flavors. Bulk ATAC gives deep coverage per region and strong transcription factor footprinting, ideal for sorted or homogeneous cells. Single-cell ATAC gives cell type-specific peaks and rare regulatory states, but the data are extremely sparse, just 0 to 2 reads per site, so peaks are usually called from pseudobulk. It helps to think of RNA and ATAC as two layers of the same biology. RNA is the output, what genes a cell is using right now. ATAC is the control, which regulatory DNA is open, the wiring behind that expression. RNA is mature and high resolution. ATAC is sparser and nearly binary. Together, they tell a fuller story. And you can measure both in the very same nucleus. That's multi-exon. Because expression and accessibility come from the same cell, you can connect a regulatory element to the gene it controls and build gene regulatory networks. It costs more, but it's worth it when regulation, not just expression, is the question. Let's put it all together as a simple map from question to essay. Which cell types and states? Single-cell RNA. Tissue-level change across many samples. Bulk RNA. Isoforms or splicing. Long-read single-cell RNA. Where are cells and in what niches? Spatial. Which enhancers and transcription factors are active? Single-cell ATAC. And to link regulation to expression? Multi-exon. A few concrete calls, using the brain as an example. To map neuron subtypes in a region, start with single-cell RNA or multi-exon if you also want regulation. To study isoform switching across development, use long-read single-cell RNA. And to understand the layered architecture of the cortex, who sits where, use spatial. Same tissue, different questions, different tools. It's also worth keeping the practical ladder in mind. Bulk RNA is the lowest cost and simplest to analyze. Short-read single-cell is moderate cost, with mature pipelines. Single-cell ATAC and spatial are higher cost and more complex. And long-read and multi-exon sit at the top, the richest readouts, but the most demanding on budget, cell number, and analysis. Finally, remember these methods are complementary, not mutually exclusive. The strongest studies layer them. Bulk RNA to scale across many samples, single-cell to define the cell types, spatial to locate them in the tissue, and multi-exon to explain the regulation. Each answers a different part of the question. To summarize, match the assay to your question, not to whatever is newest. Bulk gives you scale and depth. Single-cell gives you heterogeneity. Long-read gives you isoforms. Spatial gives you location. And ATAC and multi-exon give you regulation. Get those mappings right, and your experiment will answer the question you actually care about. Thanks for watching. Visit multi-exonacademy.com to learn more.